This is a video and podcast about the cell and the way that the cell works. Now, the cell is the unit of life. This is an animal cell. You can tell it's an animal cell because it's got no cell wall and there's no real fixed shape to it. The first thing to draw attention to is the fact that it is very much a sphere. We often look at cells as a two-dimensional image on a page, and you can see here that this is cut into, and you've got the nucleus in the middle, which is like a bit of an orange, which contains the DNA, and around the outside of the nucleus you've got a nuclear envelope, which is a double membrane, and then you've got the membrane-bound organelles, which go beyond your GCSE learning. We have here, which actually you can see is continuous with the nuclear membrane, the rough endoplasmic reticulum, and hopefully you'll be able to see some little specks on this. And they are membrane-bound ribosomes, which are responsible for protein synthesis, making proteins. And we also have here the gold colour is Golgi Apparatus, or Golgi bodies. And once the proteins have been made in the rough endoplasmic reticulum, the proteins are transported to the Golgi Apparatus where they are modified. And an example of this kind of behaviour would be we've recently learned about the fluid mosaic model with proteins in the phospholipid bilayer, and some of those proteins are things like glycoproteins, which are sugars, polysaccharides which are linked onto the proteins, and these can be used in recognition. And so the Golgi apparatus is the kind of area where the proteins might be modified and have the sugars added to them, to be then taken out to the cell membrane and expressed on the surface of the cell membrane as a receptor. Also in the cell we have the things that we are familiar with. So we have the mitochondria, which is responsible for respiration. You can see here now at A level in a little more detail, you can see that it's highly folded inside, and that's to increase the surface area. Once again, these are phospholipid bilayers, and they are membranes upon which enzyme reactions take place, which you'll learn about in respiration. We also have something like a lysosome, and a lysosome is a vesicle, and a lysosome, you can't quite see actually, it's Lysosome, is a specific vesicle. A vesicle is a lipid bilayer in a sphere which is able to separate or compartmentalize its contents. Now this would be relevant for a lysosome because it has digestive enzymes in it that are going to break down, lysis means break down, and are going to break down substances or particles which might be absorbed by the cell or are no longer needed by the cell. But this is effectively a digestive vesicle, and vesicles are used in the cell as little compartments which move around. We've got ribosomes, which you learn about in GCSE, which can be free as well as being on the rough endoplasmic reticulum where protein synthesis takes place. So there's our cell. Obviously the outside here is a plasma membrane, a lipid bilayer punctuated by proteins. It's got cholesterol in, and the proteins are used for receptors, a lot though for transportation of substances in and out that can't go through the membrane. Quick recap on that. Small non-polar substances, which is non-charged, are able to go through the lipid bilayer because they are able to interact with the lipid part, the hydrophobic part of the cell. Charged particles or bigger molecules are unable to get through the lipid bilayer, and therefore they're going to need proteins to transport them. Either channel proteins for simple diffusion through the channel, or facilitated diffusion, which happens down a concentration gradient like simple diffusion, but it involves, without energy, the carrying of proteins by, substances by carrier proteins. And then active transport, which is against a concentration gradient across the membrane, but this requires energy. So that is our cell, and we now have it in 3D form compared to the 2D form. But how does this actually work? Well, we've talked about protein synthesis here in the rough endoplasmic reticulum and the free ribosomes. So let's actually get a feel for what the cell looks like. And there's a fantastic animation which is able to show us some of the processes. Now, this here is the nuclear envelope. This is a a lipid bilayer. So these are phospholipids on the top, and these are the nuclear pores. Now, you can't see here, but below here, there would have been the formation of something called messenger RNA. Now, we all have DNA in our cells, and the DNA is the blueprint for the proteins that form us. And when we do protein synthesis in our cells, DNA is unwound by an enzyme called helicase, and one of the DNA strands is then copied into messenger RNA. And because DNA can't leave the nucleus, we need a inverted commas messenger to take the message, the code of the DNA, out into the cytoplasm where proteins can be made. A gene is a sequence of DNA that codes for a specific polypeptide, which is a protein. And so the sequence of DNA is copied into messenger RNA, and that messenger RNA flies out of the nucleus into the cytoplasm or to the rough endoplasmic reticulum, where it hooks up with a ribosome and a protein is formed. And we can see this process right now. You can see here the messenger RNA flying out into the cytoplasm, and it's going to hook up with a ribosome. This is a part of the ribosome here, and then here it goes, and then a protein is being made. You can see it going into its tertiary structure very quickly, and then that protein is going to be taken off as a metabolite to the mitochondria, as you can see there. But can you see the three-dimensional process there of the messenger RNA leaving the nucleus and going into the cytoplasm? So if we go back to our cell, here's the DNA, it's copied into messenger RNA, and it flies out of these pores, and it will find, in that video, it found a free ribosome, but it could easily attach to a ribosome that's on the rough endoplasmic reticulum. Okay, so we've got our protein made. Well, then what's going to happen? Well, I talked earlier about that protein being modified in the Golgi apparatus. So it's got to go from the rough endoplasmic reticulum to the Golgi apparatus. And how does it happen? Well, this happens in the vesicles I was talking about earlier. As I mentioned, the lysosome is a specific type of vesicle which contains a hydrolytic enzyme, digestive enzymes. But a vesicle can be any compartment of a lipid bilayer which has contents in. And this perhaps is one of the most stunning videos, because this shows how vesicles are transported around the cell. The cell has, which you can't see here, an unbelievably intricate cytoskeleton. And it is but like a spider's web or a road system flying all in and out. Proteins made of usually actin, which is a protein which is actually used in muscles. And these are tiny strands upon which, quite unbelievably, vesicles. So, for example, imagine a protein had been made and was put into a vesicle to go to the Golgi apparatus. It is taken by, these are motor proteins, and they are dragging this vesicle, which these are phospholipids, and there's some of these are probably for recognition, but it's dragging the vesicle along the cytoskeleton. If you look at that again, quite unbelievable, and that would enable these, and you can see the vesicle there, going along one there, vesicle here, and vesicles in the background, and these vesicles are constantly being taken around the cell to where they need to go to. Okay, so this, for example, if this was a vesicle of a protein that had been made in the rough endoplasmic reticulum, this is going to be taken to the Golgi apparatus. What happens at the Golgi apparatus? Well, the Golgi apparatus is also similar, you might have noticed on the other slide there, to smooth endoplasmic reticulum. It doesn't have ribosomes. And here, in the Golgi apparatus, it's quite globular. And so this is a vesicle being taken. And you can see this is the smooth endoplasmic, and parts of it bud off into vesicles themselves. And these vesicles are then taken to the plasma membrane, and they might be ejected. Here we have proteins that are the vesicles fusing with the plasma membrane, and it's ejecting its contents. This is the phospholipid bilayer, the outside of it. So these are the phosphate heads, because this will be an aqueous solution. So, recapping about the cell, first of all in 3D and then in animation. Nucleus, DNA. DNA is the blueprint for all the proteins that are going to be made, which form the characteristics of an organism. The DNA is copied into messenger RNA, and the messenger RNA is able to leave the nucleus and is decoded into proteins by ribosomes, which are either free or on endoplasmic reticulum, which makes them rough endoplasmic reticulum. The proteins then travel in vesicles along a cytoskeleton you can't see to the Golgi Apparatus, or Golgi body, where they are modified, for example, adding sugars to make a glycoprotein or fats to make glycolipids, and then they would be taken by vesicles, for example, if it was a glycoprotein, to the membrane, where it would then be expressed as a glycoprotein and ejected onto the cell surface membrane. And that is the way that a cell will work in terms of its manufacture of proteins. That process is happening thousands and thousands and thousands of times a second. Elsewhere in the cell that we know about, of course, in the mitochondria we have respiration. Centrioles here are used for cell division and the separation of cells when mitosis is happening. And other than that, this is effectively the ways that cells are now A-level. A lot of it based upon protein synthesis, and a lot of it involved in transportation of those proteins or modification of those proteins around the cell.